Lipids are a key source of fuel molecules. In healthy individuals, adipocytes, or fat cells, manage lipids. They control lipid storage in lipid droplets and lipid mobilization to provide energy to tissues in need. The amount of body fat and consequently lipid droplets needs to be maintained at a certain level. Having too few or too many lipid droplets leads to lipid storage-related pathologies. In this video, we'll focus on dysregulation that causes too few lipid droplets. Too few lipid droplets leads to pathologies including lipodystrophy and cachexia. Both are characterized by the wasting away of fat tissues. Cachexia will be the focus of a later video. Before we consider the endogenous and exogenous factors altering the balance of lipid storage and mobilization, we need to review how this dynamic equilibrium is maintained under normal conditions. To maintain an healthy lipid level, adipocytes require proper development, differentiation, and survival. These processes depend upon a number of factors. Let us first consider the differentiation of pre-adipocytes into adipocytes. This step is under the control of hormones such as insulin and cortisol. These hormones are a signal of excess nutrients and promote the formation of adipocytes. Pre-adipocytes differentiation requires the expression of two key proteins, the kinase AKT2 and the nuclear receptor PPAR-gamma. In patients with familial partial lipodystrophy, one of these two proteins is defective. In these patients, pre-adipocytes do not differentiate properly, leading to a defective lipid storage. After adipocytes maturation, proteins such as lamin A and ZMPST24 are crucial for the nuclear envelope function. Defect in any of these two proteins trigger apoptosis, the programmed cell death in adipocytes. The loss of mature adipocytes leads to a decrease of fat storage and to lipodystrophy. Finally, the adipocytes maturation is governed by lipid storage. Nutrient level, relative amount of lipogenesis and lipolysis are key factors in the maturation process. Let's take a closer look at the formation of lipid droplets and the mobilization of fatty acids stored in lipid droplets. Lipid droplets were described in a previous video, but let's review some features. They are surrounded by a lipid monolayer associated with proteins such as perilipin. The level of stored triacylglycerol inside the lipid droplets depends on the rate of triacylglycerol synthesis and the rate of mobilization by lipolysis. These two rates are tightly controlled and depends on the body's energy needs. So at high energy state, three enzymes, GPAT, AGPAT, and MGAT, catalyze the addition of fatty acids to glycerol or to monoacylglycerol to produce deacylglycerol molecules. The final fatty acid needed to form triacylglycerol is added with the help of two enzymes, GGAT1 and DGAT2. Triacylglycerol molecules start to accumulate in the membrane of the endoplasmic reticulum. Triacylglycerol will then accumulate between the two leaflets of the endoplasmic reticulum membrane, forming an oil lens inside the bilayer. As triacylglycerol continues to accumulate, the lens grows, and then the lipid droplet buds off from the endoplasmic reticulum. Genetic mutation reducing the activity of AGPAT are implicated in over 50% of congenital generalized lipodystrophy. Indeed, AGPAT deficiencies reduce the synthesis of phosphatidate and deacylglycerol, two of the triacylglycerol precursors. Now, as you recall, when energy is needed, hormones trigger the phosphorylation of perilipin. The consequences are the activation and the recruitment of specific lipases at the surface of the lipid droplets. The lipases catalyze the sequential removal of fatty acid from triacylglycerol molecules and the release of glycerol. Fatty acid bound to chaperone proteins are exported to other tissues. Fatty acids are used as fuel molecules in tissues such as muscle, while glycerol is imported to the liver, where it contributes to glycolysis and ATP production. Any mutation leading to an uncontrolled activation of lipolysis will also result in lipodystrophy. For example, some mutations prevent perilipin inhibition of the ABHD5 binding to the lipase ATGL, leading to a constitutive activation of lipolysis and ultimately to lipodystrophy. So far, all the discussed examples leading to lipodystrophy have a genetic cause. However, there is a second form of this pathology called acquired lipodystrophy. This form of lipodystrophy can be the result of an autoimmune reaction or caused by drug treatment. One known case is the HIV-associated acquired lipodystrophy. In the US, about 100,000 HIV-infected patients are affected by this form of lipodystrophy. Patients lose fat from their face, limb, and extremities, while fat deposition around the neck and the abdomen is observed. Acquired lipodystrophy develops in response to antiretroviral drug treatment. For example, nucleoside reverse transcriptase inhibitors contribute to lipodystrophy through mitochondrial toxicity. Studies also suggest that protease inhibitors inhibit a protein needed for the processing of the nucleoprotein lamin A, leading to apoptosis in adipocytes. In conclusion, lipodystrophy has a variety of causes. At a cellular level, defect in adipocyte development and function, or loss of adipocytes by apoptosis, lead to lipodystrophy. At the molecular level, triacylglycerol synthesis impairment or constitutive lipolysis also lead to lipodystrophy. There are two possible causes for these pathologies: genetic mutations or drug treatment.